Hi everybody, welcome back and start a new week and we're into chapter 7 and 8. This is all going to be about membranes, their structure, their function, their chemistry, and then membrane transport. And we'll start off pretty general and slow, and then we'll build to some relevant examples towards the end and finish up looking at some energetics and kinetics again. As you see from the slide here, it doesn't look like there's too much here, but in fact there's so much here that I couldn't fit it all on the slide. So some of these numbered sections actually have multiple parts, so sometimes there'll be many videos for multiple parts. And so what I've tried to do with this set of videos is actually make them shorter so they're a little bit more digestible for you. So there'll be more of them, but they'll be shorter hopefully. So with that, let's get going. First thing we're going to look at is some general overview on the functions of membranes to start out nice and easy. As you know from general biology, membranes define the boundaries of a cell and also its internal compartments. And so they play multiple roles during the lifetime of the cell. There are numerous roles that we can think about when we think about the cell membrane. They define the boundaries of the cell and importantly the organelles. I think a lot of introductory biology classes overplay the role of the cell membrane and ignore the role of the organelle and internal compartment membranes. So in acting as boundaries, they are obviously selectively permeable, and we'll talk about selective permeability again because it's an important principle to cement when we talk about cells and cell biology. Cell membranes, organelle membranes, also serve as sites for biological functions. For example, the electron transport chain is housed in the inner mitochondrial membrane. The bacterial cell membrane is where you find the electron transport chain in bacteria. So there are many processes which are arranged into membranes, and in many cases this increases the efficiency of that biological process. Transport is obviously an important function of the cell membrane because of this property of selective permeability. So there are transport proteins that regulate the movement of materials in and out of the cells, and also, don't forget, in and out of organelles. Membranes are sites of receiving signals, so down here number 4, receiving information from the environment, so there can be an appropriate response by the cell. And then lastly here, we can think about the role that the membrane plays in interacting with other cells in tissues. So there's cell-to-cell contact in tissue formation and development, holding cells together into tissues to form functional structures, and also those cell-cell contacts made by the membrane and by proteins in the membrane are important in communication, and so cells can communicate directly via the contacts that they make across their membrane. So let's go back to this selective permeability business again. So don't forget that the membrane is selectively permeable for 2 reasons. The first one is because it's got that hydrophobic core of fatty acids, and the second one is because each membrane has its own set of specific transport proteins which allow certain things to move and other things to not move. So as well as compartmentalizing the cell, membranes compartmentalize functions within the cell, and we usually think about those functions occurring in specific organelles. So we could talk about the membrane of the Golgi, and there are differences in the membrane of the Golgi when we look at it compared to the endoplasmic reticulum. So we'll come back to some of those things. One of the most important things we'll take a look at as we move through into chapter 8 are transport processes. Cells have to take up nutrients, obviously. They have to remove wastes, and not all of those nutrients and wastes can be freely diffused across the membrane. Some of them have to be actively transported, some of them have to be helped, even if they are diffusing. So we'll talk about some of those processes a little bit, but a lot of that I'm going to leave to you and your readings because we talked about it extensively in general biology before. So let's just think a little bit about some of these functions in a little bit more detail. Cell signaling is often a function of the membranes overlooked in introductory biology classes, so let's just talk about that momentarily. A cell receives information from its environment that it has to respond to. That could be chemical information in the form of a ligand that binds to a cell surface receptor. It could be electrical information, for example, that a cell has to respond to. And so we have this process of signal reception, which in many cases happens at the surface. And then there's a process of signal transduction, which is all this business down here, where we see receptors of some form, we come back to those in a moment, sending information into the cell, and then there being responses via either transcription or in the behavior of organelles and in the behavior of the cytoskeleton. So those would be the typical cell responses we see. So in terms of reception, we can have ligands which come directly across the membrane, lipophilic ligands. They diffuse straight across the membrane if there's a gradient. Transporters which bring in solutes which act as signaling molecules. Ion channels that change the membrane voltage. G-protein coupled receptors you will have talked about before. Enzyme linked receptors using things like tyrosine kinases. There are receptors which are associated with the cell membrane that bind to the extracellular matrix, and changes in the tension of the extracellular matrix can send mechanical signals across the cell membrane. And then there are ligands that are found on the surfaces of other cells which can bind to receptors on target cells, and when there's cell-cell protein-protein interaction is made, a signal is transduced into the cell. So that's just a little bit about cell signaling, and I'm just talking about this briefly because I think it's an important aspect that's overlooked. Another function of the membrane is really to provide the sites for proteins so that cells can be held together in multicellular organisms. So a lot of time, cell-cell contact in animal cells or in animal tissues is maintained by a group of proteins called the cadherins. And so they basically will work to hold cells together by spanning the membrane of one cell and binding to a cadherin on another cell. And these cadherins are very specific for certain cell types. So mesenchymal cells will interact via one type of cadherin. Epithelial cells will be held together by a different type of cadherin. And when these things get messed up, it can be important in processes like metastasis, which is when cells start to move around during the invasive stage of carcinogenesis. So if we think about some proteins involved in cell-cell contact, adhesion proteins form adhesion junctions. These are things like the cadherins. They hold cells together into tissues. Tight junction proteins bind membranes together and form very tight seals so that fluids can't leak across epithelial barriers, like from the GI tract, from the stomach into the body cavity. And then there are structures called gap junctions, and they allow small solutes to move back and forth directly across the two membranes of adjoining cells, and that can be for nutrient exchange or for signaling. And if you remember back to the cell biology section of general biology, the analogous structure in plants there are plasmodesmata. Those are channels across which you even find strands of endoplasmic reticulum running. So that's just a real quick overview of the functions of membranes. Lots more detail you can add to this. Next time when we come back, we'll start adding some new information, talking about lipid rafts and microdomains. I'll see you soon.